if that's something you enjoy. If not, I hope you didn't have a nice Halloween. I don't know. Okay. So if you just came in, please jump on to Socrative. We've got a quick one-question quiz going. And while you're finishing that up, I've had a whole weekend to think about chi-square or not. Does anybody have any more questions about chi-square analysis? 2 people turned in the practice, which you don't have to do. 2 people turned in the practice Google Sheets that I prepared. I gave them feedback. So that's a good way of doing it if you want feedback and practice on doing chi-square analyses. I thought I would throw this up here. Maybe it would stimulate some discussion in case there's discussion to be had. First, something that I didn't specifically go over, but it's probably useful to talk about. How do you know when to use a chi-square test and when not to? So we haven't talked about any other types of statistical analyses this semester, and we probably won't. So this is going to be the only one we're going to use this term. So if you think you need to use a statistical test at some point, it's probably the chi-square test. But these are just some basic principles for how to know whether the data you're collecting would work for a chi-square analysis. Right. Data and expected observations have to be numbers, counts, numbers of observations, whole numbers, or even decimals. They need to be categorical. That's the most important part. There need to be discrete categories of things that you can bin or assign different observations to, heads or tails, for example. Be discrete categories that all the data are assigned to. And the last one is most important, and I write this one specifically based on some of the feedback I've gotten on those example chi-square practices, is your expectation has to be based on. There has to be an expectation. There has to be some sort of expectation that follows from logic or from reason, or from your life experience. So in order to give you a more specific example of that point, I need somebody to help me with this question. All we've seen so far are examples of degrees of freedom equal 1. 2 categories, degrees of freedom is 2 minus 1 equals 1. That's all we've seen so far, but there are lots more types of analyses that don't. So can you think of an example where you would have a chi-square test you would want to use with a different number of degrees of freedom?
Where there's more than 2 outcomes?
Right, so there has to be, that's exactly, to restate the question.
For instance, like a die that has 6 sides.
Right. When is there greater than 2 bins or categories? Right, so a 6-sided dice would be 1. Actually, I guess that would be a die if there's only 1. Dice is plural, isn't that weird? Sorry, don't get me going on the English language. So, right, if you had a 6-sided dice and you wanted to know that it was fair and that you were rolling each number an equal number of times, then how many degrees of freedom would that be?
5.
You've got 6 categories. You could roll 1, 2, 3, 4, 5, or 6. So that would have 5 degrees of freedom. There are 6 different categories, 6 different outcomes, 6 different columns of data. How many times did I roll 1? How many times did I roll 2? How many times 3, and so forth. So hopefully you brought a device. If not, you can always do this later, but we're going to walk through this example right now. So if you've got a device, hopefully you're looking at text so you can copy and paste this instead of copying it. But blast this sequence, if you're capable of doing that right now, and we're going to see what gene this is. And by the way, there's one detail to add to this, which is the bottom here, against a specific database. So when you blast at NCBI, you can go down here, the bottom of this screenshot says database, and there's a drop-down menu. So it defaults to a particular database. I'm suggesting for this specific case, you want to paste that sequence up here in the query sequence dialog box, and make sure you choose the others database, and then choose nucleotide collection, NR/NT. It's a subset of all of the DNA sequences that are in the whole database, in this sub-database, the NR/NT database. NR stands for non-redundant. It means there's one example the curators of this database have put one example of every gene into this small database, NR, non-redundant. It means there aren't multiple copies of each gene in this version of the database. Because it's a smaller database, it's easier to search through. Does anybody have any technical questions or concerns about blasting? So if everything works right, you can copy that sequence, paste it into this website at this URL. And if you get that far, and if not, let's talk after class or office hours. I'll help you individually work through this process if you want. These are the questions to answer, just based on this one BLAST search. What's the gene that that DNA sequence is part of? What organism is it from? What's its accession number? That is, what's its unique identifying record in the database? What's its DNA sequence, and how many nucleotides long is this gene that this particular sequence is from? It's a tiny little bit of sequence from a bigger gene. Have we broken the Wi-Fi yet? So if you did what I did, which you may or may not have, you'll see a screen that looks something like this. And this contains most of that information that we're looking for, at least to start with. So here's the top list of hits, matches. And they're sorted from top to bottom in order of how good a match, according to the calculations that the database does to assign scores to the different matches. So that was our query sequence, whatever DNA sequence, we don't know what it is, we pasted it into the search. What are these sequences? These aren't queries, these are the other type of sequence. There's queries and there's subject. So this is a list of subjects. NCBI's version of search results, same thing. So what's the best matching subject? What's the best record entry DNA sequence in the database?
Chromosome number 20 for eukaryotic cells.
Eukaryotic synthetic construct, chromosome 20, whatever that is. I don't know. If you want to know, click on the name. It's a link, and it'll tell you details about what the hell that means. Okay, next one down. Homo sapiens prion protein. Has anybody heard about prions?
Oh, yeah.
Okay, some have not, maybe. We'll talk about prions a bit more later, especially when we talk about protein folding. So we're almost there. We're going to do this, then transcription, then translation. Prions are the things that cause, what are the various diseases they cause? Mad cow disease, scrapie. So mad cow is the cow version, scrapie is the sheep version, Creutzfeldt-Jakob is the human version. It's a disease that basically more or less eats away at your brain and is caused by this gene. So, I don't know, it's not my favorite gene, but it's an interesting gene to learn about. So that is where this DNA sequence came from. The reason that I conclude that is because most of the top hits, most of the highest scoring subject matches are to the same thing. Prion protein, and then a genomic clone, and then another prion protein. So if you click on, for example, there's the accession number of the gene. So that's another answer. What was the gene, prion protein? What species is it from? Homo sapiens, right there. What's its accession number? Right there. The main reason you want to know about the accession number is if you're communicating with another geneticist about a gene that you're doing some analysis on, you get this DNA sequence and you study it, it's useful to be able to tell them, I was using this specific version of that gene that's in this database. So instead of just copying, pasting the DNA sequence and emailing it to the collaborator, you just say, I'm using this accession number. And they can look up all the data themselves if they want to. What else do we have? So if you click on that link, it brings up this new page that's got, again, the accession number. What happens if you click there?
you get the whole entire sequence.
You get the FASTA sequence. So if you want to get just the DNA sequence, that's it. Click on the FASTA link. It's got the length of the gene, or at least the length of the number of base pairs in that FASTA sequence, 22,438. It's on chromosome 20 in humans, and again, it's from humans. So we get a lot of information in a pretty condensed way. So this is an easy way, if somebody hands you a DNA sequence, they say, what is this? This is a tool that a lot of geneticists will go to figure out, what is this gene that I'm looking at? DNA sequence do. And at the start of class, I created yet another set of exercises on this whole set of topics using BLAST, identifying genes, and it's now to you on Google Classroom, so you can do that whenever you want. I'll post a key later this week. Not for points, just for practice, I'll post a key. Any questions about why use BLAST, what BLAST does, how to use it? There will be a lot of practice opportunities, so you'll get more chances to ask me questions about details if you like. All right. So moving back into the gene, this is probably our last day talking about the gene, and then we're going to move on to transcription. So how the cell knows where the genes are on the chromosomes and how it knows what to transcribe and turn into messenger RNAs or other sorts of RNAs. But first, here's an example gene, SRY. Brought it up the last couple of classes. It's the gene that causes male sex in humans. And this was our case study for today. We already knew something before this paper came along, 1991, so that, what year is it? So that probably predates many of you. This was a really outstanding and powerful paper. This is the first time that a sex-determining gene was functionally studied. And what they knew before they started this study about SRY was that you could find, there were humans, and this is one of the nice things about working with humans, is humans go to the doctor when they sense that something is amiss. Most animals don't do this, but humans do. So we have a lot more information about medical issues with humans because we report them to doctors, and doctors report them to scientists, and then the scientists study them. We knew that there were XX individuals that had SRY, or was it the opposite? There are two different types of studies you can do. What sort of a sex would an individual be if they had two X chromosomes? What sort of phenotypic sex would an individual be with two X chromosomes that contained SRY? Because the only gene, male, because the only gene on the Y chromosome that's essential for sex determination is SRY. That's what this study proved. So that's what this study is going to look at, is what happens when you add SRY to an XX mouse, not human, in this study, as you can see in the title. But before the scientists did this study, they knew something else too. And they knew that XY individuals had been identified that had mutations. So the Y chromosome has SRY, but some people have SRY that has a mutation. I just denote that with an asterisk. And these individuals are phenotypically female. Which was the first indication that this gene was involved in sex determination. Because if you have a mutation in it that maybe makes this protein not work, then you don't develop as a male. So it implicates this gene in the importance of creating males. So there are two different sorts in these two different colors that I've drawn up here. There are two different types of genetic analyses that are important for you to know about when scientists study the roles of genes in creating traits. The bottom one tells us something about SRY. It's an adjective, this word that I'm going to be looking for. if you don't have a functioning version of SRY and you can't be male, or you don't develop as male, then what word would you use? Fill in the blank. SRY is, fill in the word, for producing males. Essential, necessary. Same idea. You have to have it. So this is the concept of necessity. And that's the sort of experiment that geneticists do to test whether a gene is necessary to have an effect. If it's necessary to do something, then you get rid of the gene. You mutate it, you delete it, you remove it from the individual, or you look for a naturally occurring mutation. And if that mutation causes whatever trait it is to disappear, like maleness, doesn't happen, then you know that SRY is necessary to produce males. This other experiment that these authors did in this 1991 paper, the one in blue up here, they had mice that were female, genetically female. They add SRY. And this is a couple slides down from where we are, but on a roll. So I'll keep on this slide. What does that test about? What was their expectation? What did the authors expect? They add this one gene to an XX mouse. What do you think is going to happen?
They would expect the mouse to be phenotypically male.
Right. So if SRY is something that triggers male development, then if you put that gene into a mouse that doesn't have a Y chromosome, if SRY is all you need, then it should become male. That was their prediction. That was what this whole paper was about. So that test, moving a gene from an individual that has it to an individual that doesn't, this is cisgenesis. It's like transgenesis. You're moving a gene from one organism to another organism. We call it cisgenesis in this case because they used a mouse gene to put in another mouse. So because it's in the same species, we call it cisgenesis. Keep the genes within the same species, it's cis. If it's a transgenic organism, which we'll talk about later when we talk about GMOs, transgenics are when you move genes from one species into a totally different species, like put bacterial genes in plants or something like that. That's transgenesis. This is cisgenesis. Take a mouse gene that's normally found in males, put it in an XX animal, see what they do. Did they develop as females or males? So here's the other word. We've talked about necessity. You get rid of the gene, see if the trait disappears. Test if that gene is necessary to produce that trait. This other experimental approach tests whether a gene is sufficient to produce a trait. What's another way to say sufficient? If something is sufficient? Enough. That's all you need. If this one gene is sufficient to produce males, that means it's the only thing that you need to become a male. That would define it as the master regulator of sex determination. It's a switch, a type of switch. If you have it, it can turn on, flip all the downstream switches. It can turn on all the other genes that you need to produce a male. So, necessity and sufficiency. Move the gene and lose the gene. That is, you move it from an organism that has it to another organism that doesn't and see what happens to that other organism, and you can delete the gene, get rid of it, and see what happens to the organism. Necessity and sufficiency. Okay, so we've already talked about SRY and what it does. Don't need to cover that again, I don't think. And it's on the Y chromosome in both the human and the mouse genomes. Human and mice are both mammals. We both have the same Y chromosome. They're different, but they share this gene, SRY. So it's found on the Y chromosome in males. It's not ever found in phenotypic females. Although I suppose it could be. What would be true about SRY if you found that gene in a phenotypically female individual? Well, but it's female already, so that's, yeah, exactly. Maybe that is a mutant SRY gene. So if you put a non-functional SRY gene we would still say that the SRY gene is there. It's just a mutant version. If that mutant version doesn't have the capability of turning on male sex determination, you could technically be XX and have SRY and still be phenotypically female. To my knowledge, that's never actually been discovered in humans, but never say never. By the way, so this is one of the figures from this paper, and we're looking at two different regions of chromosomes. So chromosome represented as a black horizontal line. This is the mouse version of the Y chromosome where this gene, SRY, is located. That's that gray box above the line. And here's the human version, same part of the Y chromosome. What are the E's and S's and H's? Anybody pick that up? Those are restriction enzyme sites. E stands, in this case, E stands for EcoR1, which is a very commonly used restriction enzyme. So if you were trying to, and this is what these authors were doing, if they wanted to isolate the SRY gene from a mouse, male mouse, and put it into an XX, presumably female mouse, if you cut up this part of the Y chromosome and you electrophoresed all those fragments, what size piece of DNA would you be looking for to add to the females? So if you digested all of that with EcoR1 and electrophoresed it, so you saw the different band sizes from that piece of the Y chromosome, which band size do you want that contains this gene?
3.5.
Yeah, so this gene is flanked on two sides by EcoR1 sites, and that product is 3.5. In this case, it's 3.5 kilobases, or 3,500 base pairs. So basically what they do, they cut out this gene, and they microinject it into female embryos. Sorry, not female embryos, embryos. So we've already covered all of these things. Summarize. The hypothesis, SRY is, let's see, what was the thing that hadn't been proven yet? It was necessary for maleness or sufficient for maleness? See if it's, that was already shown. That was the humans that had mutant versions of SRY that developed as females. So if you didn't have it, you weren't male. So that attested necessity for it. So this one, indeed.
So the hypothesis is that it's necessary?
Is it sufficient or is it necessary? That's my question right now.
Okay.
Right. So the logic again. If it's necessary, then if you get rid of it, you don't see the trait. That was what was shown earlier. So if it's sufficient, if SRY is the only thing that's necessary to make a male, if we, what was their main experimental approach?
Injecting SRY into embryos.
Yeah, put SRY in an XX mouse. Then they expect, we already talked about this, they expect that XX organism to become a male, or to develop into a male phenotypic male. That was the goal. That was what they did. Technically really difficult. Isolate some DNA from mouse chromosome that has the gene that you want. Collect usually a single cell or a few cell embryo, a bunch of them, from female mice. Use a tiny microinjection needle, inject this DNA into all of these embryos, and hope that in some of those embryos, that DNA you injected into the cell gets taken up by the nucleus and added to the nuclear DNA. That is, the DNA they inject has to be incorporated into a chromosome. So they have a microinjection needle with little fragments red of the SRY gene. They hope that that DNA gets into the nucleus and actually adds itself to one of the chromosomes that are already in the nucleus, because that's the only way that gene, SRY, is going to get passed from cell to cell, so that every cell in this maturing, developing organism has that gene so that it can do its job, or do what its job would be if it was in an XY mouse. So technically really difficult. They injected lots of mouse embryos. I think I have the data here, just a second. No, we'll get to it eventually. I do have that data. So if you're looking for where to find this information, so 5 minutes. If you want some information about reading primary literature and how to quickly find information in primary literature, research studies, please pay attention. If not, please work silently doing something else. As always. So where in the primary literature, primary research literature means that somebody, a scientist who's actually conducted experiments and they put the data in this paper and they're trying to share this data with everybody. That's primary. That's basically contrasts with review papers, which are papers that, manuscripts, articles, that don't contain new data in them. They're just an author summarizing a bunch of other previous studies. Those are called reviews. That's sort of the main distinction. Primary research articles, reviews. So this is a primary research article. It's got the raw data in it. Hopefully you looked at some of it. There are a couple places to find these points: the hypothesis or the goal of the study, the approach the authors used, and then what they expected to find. All of those things are usually found in the abstract, which is usually the first part, top of the first page, or in this case, the upper left of the first page. Now, sometimes it's difficult. So in this case, the hypothesis or goal was this recently isolated gene has many of the genetic and biological properties expected of a sex-determining gene. So we read into that, well, expected, which means that no one's showed that yet. So that's going to be the goal of this paper, is to address that point. And then they state their conclusion a little bit. This DNA fragment is, oh look, there's that word again, sufficient to induce testis differentiation when introduced into female mouse embryos. So that's what they did. And that's what they found. Sufficient to induce testis formation in an XX mouse. So that little paragraph has what's the goal, what did we do, and what did we find. Those same details are also elsewhere in the introduction. Normally, if you want the cheat sheet to reading primary literature and introductions, the big picture stuff, which usually includes what we call a gap in knowledge, that is, what don't we know? Somewhere in here, the scientists are going to try to say, we did this really cool thing because nobody had ever done this before. We usually refer to as a gap in knowledge. What's not known? What's the big idea? What's the question that we're trying to answer? That's usually at the start somewhere. And then when we get down to the end of the introduction section, which is the first section in the paper, that's usually results and just a summary. One paragraph usually the last paragraph in the introduction has all the information. What we did, they usually restate. What did we do? What did we find? What was the conclusion? Results and interpretation. And then, of course, if you want to, you can go read the other 9 pages, where you get all the details, the materials and methods. So if you wanted to replicate the study, you could do it yourself. All of the data, the results section, and then the full interpretation of all of the data, which is the discussion section. Okay. The introduction's there at the start for a purpose, so start with that. Does anybody have any questions about anything about primary literature? Reading papers, structure of papers. We're going to do this several more times before the end of the semester. Just for practice. Good to get used to it. Okay. What about results and interpretation? So I asked you to look primarily at this figure, figure 3. Where are you going to look to find out the information, or where did you look, if you did, to find out the information about what's going on in figure 3? What places in the manuscript did you look? To try to figure out what was going on.
caption of the image?
Okay, so that's always the first place to start. Look at the caption. Each of these, most papers nowadays, back in my day, the papers only had one figure. Most of the papers these days have lots of different panels. These subsections of a single figure, they're usually lettered, A, B, C in this case. So if you want to know the details of what's in each of those are called out specifically in the caption. That's where to look for what is A, what is B, what is C. And we refer to those as panels. Panel A, panel B, panel C. So that's the great first place to look. If you want to know what's in the figure, look at the caption. That's why it's there. Where else do you look for information? If you want to know figure 3, what's going on in figure 3, panel B? What's the fastest way to get that information? Look at the caption, look at the picture, try to figure out what's going on. So what's panel A? Socratic quiz. What's panel A? Yeah, it's gel electrophoresis of a PCR. Okay, what's panel B? Mice. Yeah, mouse testes. What's panel C? Right, this is probably the one that's least obvious. That's fine. It's actually histological sections of mouse testis. So it's looking at the cellular structure of the testes of mice. Where do you get that? You don't find it in the caption. Look for it in the text, but especially this PDF it wouldn't work on, but modern PDFs, do a search. If you want to know what's in figure 3A, type fig 3A into the search bar and find out where in the text it specifically talks about that figure. There you'll get the full information, not just the little tiny blurb that's in the caption. So one of the transgenic mice had no Y chromosome as determined by PCR analysis, figure 3A. So that gives you, maybe, if you're interested in it, it gives you a sense for what you should be looking for when you look at the gel in figure 3A. Oh, I'm trying to figure out how they used PCR to prove that mouse 33.13 had no Y chromosome. So then you go to that panel, you look at the PCR for that mouse, 33.13, and you try to figure out what do these bands mean and why do they mean that that mouse doesn't have a Y chromosome. And likewise, externally male. So when you've got the mouse doing the clasp test where they're hanging on the bar and their genitals are obvious, that's how they sex the mice. That's how they tell if they're phenotypically male or female. So you can go look, was externally male, figure 3B. So you go stare at the mouse genitalia in figure 3B and you convince yourself that it's a male. If you know anything about mouse genitalia, which I don't. Maybe you do. Make no assumptions. We've talked about this before. What are two different ways to define sex that a biologist would use to define sex?
Gamete size and ability.
Okay, gamete size.
The ability to reproduce.
Okay,
and
so
the role of reproduction.
So traits, gamete size, male, small mobile gametes, female, large immobile gametes, mating capability, and the role in mating. So those are both phenotypes. What about genetics? How does a geneticist dictate or identify the genetic sex of individuals?
Their chromosomes and the presence or absence of sex-controlling genes.
Okay. So we've got these two concepts. There's genetic sex, XX or XY in mice and in humans, and there's phenotypic sex. What did you develop as physically, in this case? So in this table, they're looking at the left column, just number of embryos. Now I guess I better actually find this information. How many? They injected 158 embryos. So one of the poor graduate students sat in front of a microscope for hours upon hours trying to stick a tiny glass needle inside 158 different mouse embryos to inject this SRY gene and hope that gene gets incorporated by the genome, the chromosomes, and passed to all the other cells as the embryo develops. And of those embryos, now they're just classifying how many of different embryos showed different patterns of these different columns. So we're going to skip sex chromatin for now. Ask me about it later if you want to know what that means. What's SRY? That's the gene. This is a particular version of SRY gene. Who didn't minus, doesn't have it? 63 embryos didn't have SRY. They also, 27 of those 63 also minus didn't have this other gene, ZFY. This is really critical. On the Y chromosome, there are multiple genes, not just SRY. SRY is one of the genes. There's another gene called ZFY. Guess what Y means? It's on the Y chromosome. Guess what ZF means? Doesn't matter. It's another gene on the Y chromosome. So mice, the 63 mice that didn't have SRY and either didn't have ZFY, that's the minus, or ND is not determined, they didn't bother looking. So no SRY, no ZFY, deduced karyotype. You know what a karyotype is. What does it mean that it's deduced? What does it mean if a detective deduces something? Right. They didn't get specific data on this. They deduced it. They used all other evidence to assume then that this was the karyotype. So what evidence suggests that these 63 embryos are XX? They don't have SRY, and they don't have another gene that's on the Y chromosome. So if they don't have SRY and they don't have ZFY, they probably don't have a Y chromosome. Therefore, they must be, if they don't have Y, the deduced karyotype is, okay, they must be XX. In other words, absence of those two genes, SRY and ZFY, suggests the absence of the Y chromosome. If you don't have the two of the genes that are on the Y chromosome, you probably don't have the Y chromosome. You're probably XX. So these were control mice, these 63. They weren't transgenic or cisgenic in my language. They didn't inject these mice. These were just regular embryos they let come to term. They phenotyped them for sex, and they were phenotypically female. It's a control experiment. No injection, no treatment. Okay, what about these 27 embryos? With SRY and with ZFY, what's their sex chromosome constitution? What's their karyotype? They have to have an X because all mammals have an X. The question is it a second X or is it a Y? If they've got ZFY and SRY, then there's a Y chromosome in those organisms, XY. Also not transgenic, also male. So the top two lines are just normal mice. No anything going on. So let's skip the next three lines. We could if we want to, but there's other stuff that's more interesting to talk about. Let's go down to the bottom line. So help me read this. These were transgenic. This is the only thing I'm going to tell you. You're going to have to do the rest of this table, this bottom line. Is transgenic. That is, these are embryos that they did inject with DNA, SRY. The problem with doing these injections is that you don't know what sex you're injecting. It's just a tiny cell. So you could be, half the time, you're probably injecting SRY into an XY embryo that already has SRY. The other half of the time, you're injecting XX embryos. And we only care about the XX embryos, because if you inject SRY into a mouse that already has SRY, nothing happens. They just become male, but they were going to become male anyway. So these were phenotypically male. They were injected. So how do we know if these were XX injectees or XY injectees? The data's up here. What about ZFY? Not there. So absence of ZFY means that these two embryos did not have a Y chromosome. Why can't we use the presence of SRY to do this? Right, we're artificially adding SRY, so it's not enough just to say, do the mice have SRY? therefore they have a Y chromosome. Because if you inject a mouse with SRY, it's going to have it. You can't use that to tell if there's a Y chromosome or not. You have to use ZFY, which is independent of SRY, to tell if there's a Y chromosome or not. So, no Y, therefore XX, but, exclamation point, exclamation point, exclamation point, XX mouse with SRY develops as a phenotypic male. 2 of them. So of the however many 741 transgenic embryos, 2 that they injected SRY into developed as phenotypic males, but they had 2 X chromosomes.
Did the study talk about any that developed both?
Not that I saw.
Okay, that's interesting.
But they did look at, in that previous panel, they looked at this. So there was something weird about this, or odd, different, I guess, about this mouse. These are both males. 33.17 was a control male. So these are, again, those testis tissue cross-sections. The male on the left is fertile. The spermatiferous tubules are full of developing sperm. Whereas the sterile male on the right, those same tubules are empty. Those are not generating mature sperm. And this happened to be that XX male. So SRY was enough to help testes develop, but the mouse was still sterile. So not really a fully functional phenotypic male, wasn't able to have kids of his own, didn't produce functional sperm. But SRY still was enough to make the male develop, or make the animal develop otherwise phenotypically. What was missing? There are other genes on the Y chromosome that control spermatogenesis. You have to have those also if you want to be fertile. So if they had injected those genes also along with SRY, then maybe they would have created a fertile XX male. But for 1991, this was pretty good in itself. It's true. So this again is the comparison of the wild type and the XX transgenic mouse, both of which, externally anyway, phenotypically male. So as far as anybody could tell at the time anyway, the phenotypically, or the XX transgenic containing SRY was practically indistinguishable, other than that histology of the testis. All right, so here's that PCR gel. So what's the evidence we have that 33.13, the individual on the right, is XX? We don't know male. Male we just saw. That was the picture. Photographs of mice hanging on a bar. That's how they got these data, the phenotypic sex. So how does this gel show us that this individual, 33.13, has no Y chromosome? Which mice have SRY? Yeah, we're looking at that band size. So that mouse, 3317, has the gene SRY. But that's a wild type mouse, so it has a Y chromosome, so that's the SRY that's on his Y chromosome. Where did this SRY come from? This was the injected transgene, cisgene. So again, that's the XX, would have been female had they not injected the embryo with this gene and created a phenotypic male. So last point for this then, how do we know that that individual doesn't have a Y chromosome? There's no band there for ZFY, but there is one in a normal male, an XY male. So that's that presence or absence of the Y chromosome. Y chromosome there, no Y chromosome there. Have any questions about this figure? Or the mouse sex determination story, for that matter? The whole point of this story really is to show one example of how a gene can be proven to be necessary and sufficient. We didn't prove the necessity part, we proved the sufficiency part. And these are the types of studies that a lot of geneticists do to study a gene, which is where we are in this topic right now. The gene. SRY is an example. So that's the gene. We're moving on to transcription and translation. And the fail exercise, first attempt in learning, was to look up information about a protein sequence. In this case, one that spells "have a nice day" in the single letter amino acid code. So the key to this is on the next slide. If you've tried it, then you can check your answers against this key. Sorry, 2 slides. If you haven't seen this before, use this. Bookmark this slide. We're going to use it a little bit, at least. The 20 naturally occurring amino acids. It's the sum of these 20 amino acids in various combinations, just like combinations of A's, C's, G's, and T's make up chromosome sequences. These make up proteins. Amino acids linked together make proteins. So this is what I use to answer all of those questions. So for G, for example, have a nice day. That doesn't have a G in it, does it? What's the letter that's in have a nice day? A. A, 3-letter code alanine, or ALA, full name alanine. And then if you want to look up the chemical characteristics, they're written up here in red. Circular, acidic, amides, basic, and amino acids. So we'll talk more about the amino acids. Remember, one key takeaway, if you haven't already heard this in organic chemistry or biochemistry, that every amino acid has an amino group on it and a carboxylic acid group, and lo and behold, that's why we happen to call these amino acids. So every amino acid has an N and a C group, and that's polarity for amino acid nomenclature. When you link these together by condensation reactions between so that condensation reaction releases an H2O and forms a bond between the nitrogen and the carbon, forms what's that bond called? Peptide bond. This is a polypeptide, multiple amino acids connected together covalently. It's a peptide bond. So those peptide bonds link together different amino acids into larger groups that are called polypeptides, and eventually we call polypeptides proteins. And each one of them then will have an amino end, and in this case, once we link those 2 amino acids together, there's the carboxy terminus, or the C terminus. C terminus. In a little while. So there's the key to that. Of course it's fake, I made it up. There's no protein that spells out "have a nice day," or at least not that I know of. So let's finish with a couple of these questions. Transcription. Is it true or false, I asked these last class, is it true or false that the 5' end of a transcript corresponds to the C terminus of a protein? So does the 5' end of the transcript, 3' end of a transcript, after translation, and this is just looking into the future, after translation, the 5' end of the messenger RNA encodes the first bit of the protein, the amino end, and the 3' end ends with that carboxy group. So this could be multiple amino acids, but the amino end of one is at the left side. In this case, it corresponds to the 5' end of the messenger RNA. Finally, well, I guess we've already touched upon this. Number 4, which of these answers is correct? Which, if any, or multiple? I'll give you a start. It's not 5' and 3'. That's DNA and RNA. So N and C works. So when I did this last time, there was a mixture of A and C. Most people said C, but some of you also said A. So I did want to have a quick discussion about that. It's not A and B. What's true about the N and the C termini that might make you think that it's plus and minus?
Because of the, N is usually positive and.
So in the case, in some physiological conditions, that amino group could be protonated and have a positive charge, and the carboxy terminus could be deprotonated and have it's an acid. The NH3 is a base, COO, carboxylic acid. So yeah, I'd give you a plus and minus, only if that's what you were thinking. Okay, so that will be the end to the gene, and for next class, we're on to transcription. So there's a case study to look at for that one, and one micro lecture video. Feel free to take a look at the OpenStax readings if you want as well.